And so we'll pick that up today, and we'll define the neuron doctrine, of course, as the neuron is the fundamental structural and functional unit of the nervous system, and we'll spell that out. But one of the things I want to describe here today is how we got to that point, how we got to the idea of the concept of the neuron doctrine, because we didn't always understand that the nervous system was made of these individual neurons. There were two major players in determining this story that I want to emphasize. There were many people who contributed to this, but there are two major players as part of this argument. And one of them is a guy named Santiago Ramon y Cajal. And Santiago Ramon y Cajal was a Spanish anatomist. And he was working in Spain, mostly in Madrid and Valencia. And he was using a technique called the Golgi Stain that had been worked out in Italy by a fellow named Camillo Golgi. So Camillo Golgi develops this clever stain that I'll tell you a little bit about that now allows people to see whole individual neurons in sections of brain. And from this, there grew an argument. And the argument was basically the cellularists who held that the nervous system is generated from individual cells called neurons. And these are separate and distinct structures. And the reticularists who said that there are neurons in the nervous system for sure, but they're all connected one to another through cytoplasmic bridges. So there's no distinction of one neuron to the next. They're all cytoplasmically the same. And so at this point, at the turn of the 20th century, there's this argument going on, whether or not the nervous system is constructed from individual cells or from a syncytium of cells. Now, to take the drama out of it, what I'm going to tell you is that Cajal figured it out. He was a cellularist. And he described how the nervous system was generated from individual cells. And from that, he described something known as the structural description of a neuron. He said neurons are constructed from a dendrite, a cell body, an axon, and an axon terminal. And in addition to this anatomic description, he provided us a level of description called the law of dynamic polarization, saying information flows in one direction. It flows from the dendrite to the cell body to the axon terminal. The reticularists, which included Camillo Golgi himself, really had no good explanation in this syncytium how information would flow only in one direction. It was a real weakness of the reticularist theory, and we'll talk a little bit about that. But we're going to describe neurons as an individual cell, and we're going to describe them from 2 points of view. One is the view that Cajal had in 1900, and that's the dendrite cell body axon. But then, as we began to understand the distribution of ion channels in these cells, we could now think about them more functionally. And that brought about a functional description of the neurons. And the functional description of the neurons is very similar in many ways to the anatomic description, but we provide some other names. The dendrite in the cell body becomes the input region. The axon becomes the conductal region, and the axon terminal becomes the output region. Now, because of the advent of electrophysiology in the mid part of the 20th century, they could now begin to describe this functional organization of the neuron in terms of which ion channels were distributed in which region. Now, Cajal and his cohorts had no idea about ion channels. That took many more years to sort out. And one of the ideas that we'll drive home here, and we'll come back to over and over again, is that the input region is characterized by a kind of channel called a ligand-gated channel. These are going to be channels that are opened by neurotransmitter. The conductal region is characterized by a kind of channel called a voltage-gated channel. So these channels in the input region are opened by a neurotransmitter. These channels in the conductal region are opened by charge or voltage. These channels here in the input region, the size of the signal generated by these channels is determined by the number of neurotransmitter molecules. They're graded over a broad range, so they're variable. The voltage-gated channels in the conductor region generate signals that are all or none. These are called action potentials, and we'll talk about all of this in great detail. But for now, recognize that when we talk about the functional description of the neuron, we have an input region where channels are activated by a neurotransmitter. We have a conductor region where channels are activated by charge, and an output region where we'll release neurotransmitter that will then impinge upon the next cell. And we'll walk our way through that. Then we'll talk a little bit about the cell biology of neurons. And we've touched on this. We touched on this when we talked about kinesin and dynein and microtubules. We'll reiterate this a little bit. And we'll bring home again the idea that with this organization, either anatomic or functional, of a neuron, we recognize a highly asymmetric cell. It's highly asymmetric in shape, and what's more, it's highly asymmetric in its function, in the sense that the asymmetric cell has all of its proteins generated in the cell body. So you have this small cell body, no more than 50 micrometers in diameter, that may have an axon attached to it that extends for a meter. But all the protein in that cell is generated in that tiny region called the cell body. That generates a logistical problem because we have to move those proteins from place to place. And there are going to be two major ways of moving the protein. One is going to be via fast axoplasmic transport at 200 to 400 millimeters a day. These are the ones we talked about. We talked about these proteins as an extension of packaging proteins in vesicles, putting them on microtubules, and moving them long distances. This is fast axoplasmic transport. And these proteins are all related to the proteins, or are in fact proteins, that have been synthesized on rough endoplasmic reticulum. But of course, we know that all cells contain not only those proteins synthesized on rough endoplasmic reticulum, but also those that are synthesized in the cytoplasm on free ribosomes. The same logistical problem exists. So now how are we going to move those proteins produced in the cytoplasm over long distances? And we'll see here that these proteins, the cytoplasmic proteins, are propagated at rates of 0.5 to 2 millimeters a day. Now, at this point, one of the points I will emphasize, one of the ones I want you to hold on to, is that signaling in neurons comes in two major places. This signaling is in the cytoplasm. This is all movement of proteins in the cytoplasm. This signaling that we'll talk about in great detail mostly next week is all at the membrane level. So this signaling of ion channels is across the membrane and along the membrane. This signaling, which is the movement of proteins, is all in the cytoplasm. So there's this very narrow shell around all of these neurons, which is the plasma membrane, only about 6 to 7 nanometers in diameter or in thickness. Then the rest of the cell, which may be a micron or more in diameter, is all filled with cytoplasm. Don't confuse the signaling between the two. One set of signaling is going to be by ion channels along the membrane, and the movement of ion channels through the membrane, and the other is going to be the movement of cytoplasm within those membrane bounds. And we'll talk about that. But let's start here with the description of the Neuron Doctrine. The Neuron Doctrine says that the neuron is the fundamental structural and functional unit of the nervous system. This is really the basis of all of our thinking in the nervous system, even to this day. Now, when we talk about glia, probably tomorrow, we'll see that the glia probably also play an important role. And as time goes on, we'll see the glia playing ever more important roles as we discover more about them. But for the most part, when we think about the nervous system, we think about the signaling by this individual unit called the neuron. But the concept of the Neuron Doctrine arose only at the turn of the last century, between about 1895 and 1905. And the Neuron Doctrine arose from the work primarily of two scientists. One whose name is Emilio Golgi. And the other is a guy named Santiago Ramon y Cajal. Cajal's a pretty interesting character. Cajal is largely thought by most neurobiologists to be the founder of modern neurobiology. As a young man, Cajal wanted to be an artist. He said, I want to express myself as an artist. His father said, go do something useful, become a doctor. Some things never change. And so he took his father's advice, and he went to medical school, and he became a doctor and then became a famous anatomist. But his art always came throughout his work, and if you put your phone away, I'd appreciate it. And his art came through in his work, and we'll see that throughout most of the 20th century, the best images we had of neurons. In fact, until the late 60s, when they could begin injecting neurons with dyes, these were the very best images we had of neurons, was his work. Emilio Golgi, his claim to fame here was that he invented something known as the Golgi Stain. Emilio Golgi was an outstanding microscopist. He understood the physics of microscopes, he understood how to use them, and in fact, in the early days of microscopy, he was the one who described the structure called the Golgi apparatus. That's why we named the Golgi apparatus the Golgi apparatus. He wasn't sure what he was looking at, and his images weren't all that clear, but he was such a good microscopist, he said, I think there's something there. There's these structures I see from cell to cell, and it became known as the Golgi apparatus. But in addition to that, he developed a new kind of stain. Up to this point in histology, the stains were such that they would stain, they were mostly acid stains and base stains, but what they would do in the nervous system is they would stain cell bodies. So they could look at sections through the brain and they could see cell bodies, but the problem was when they looked at the billion or so or more, 10 billion neurons in the central nervous system, they just saw all of these cell bodies. They couldn't make any sense out of it. Then the development of the Golgi stain, which uses chromium and silver salts. We still don't know entirely how this works, but the chromium and silver salts would label neurons. So they would get a specific labeling of neurons, but the label would only be to 1 to 2% of the neurons. Now, you would think by looking at this that this would be a terrible stain because you're only labeling 1 in 50 or 1 in 100 of the neurons. But the advantage of the system was that first it labeled 1 to 2% of the neurons, but it labeled them in the entirety, so they could recognize a neuron in its entirety. So they saw the whole cell. So now they could cut sections through the brain, and they could now see individual cells, and they could see them in their entirety. But because not all of the cells were labeled, you could see only 1 in 100 or so, you could recognize individual cells in the sections. This is a tremendous advantage. Now, Santiago Ramon y Cajal, working in Valencia and then later in Madrid, modified this stain. He took the Golgi stain and he fiddled with it a little bit. And he discovered that by modifying the Golgi stain, he could get ever more clarity of identifying individual neurons. And what Cajal did, and perhaps it was fortunate that he was working in Spain, which is really out at the outer fringe of the academic universe, whereas Golgi was working in Italy, right at the center of the academic universe. But Cajal, by working by himself and thinking about these problems for himself, really was the one who discovered the idea of the cellularists, the cellular basis of the nervous system. Now, oddly enough, Golgi, using the Golgi stain, came to the conclusion that every neuron is connected to every other neuron. By cytoplasmic bridges. They fundamentally said that the nervous system is a syncytium or a reticulum. So the school of thought from Golgi was that the nervous system is a reticulum, and they got the name the reticularists. Meanwhile, Cajal, working on his own in Spain using the same techniques, comes to an entirely different conclusion. And part of this may be the way he did his experiments. The story is that Cajal would spend the mornings in the lab, and he would sit down at the microscope at the lab, and he would look at slide after slide after slide. So those slides that he had prepared the day before, sections through brains that he had prepared and then mounted on slides, he would just sit down and look at hour after hour after hour. And then in the afternoon, he'd walk away from his lab, he'd go out into the park. The story is he would feed the pigeons in the park. And then in the evening, he would come back to the lab and he would make drawings of what he'd seen in the morning. He wouldn't sit down and trace those cells that he saw. He would draw them from memory. And so presumably what he was doing is he was taking a kind of composite or an average of the cells he'd looked at in the morning and drew them as a composite neuron in the evening. And again, as I was saying earlier, until the mid-20th century, these were the best images we had of neurons. Well, then it was around, I think, 1900, 1902, something like that, that Cajal basically went on tour. And he visited all the major universities in Europe, and he presented his material. And through the presentation of that material, at the various academic institutions in Germany and Italy and in England, he basically turned the tide. And when people looked at his data, it was so compelling that they concluded that, sure enough, the nervous system must be constructed from individual neurons, separate cells. But Golgi never accepted this. And in fact, in 1906, Golgi and Cajal, for their contributions to understanding the nervous system, were awarded the Nobel Prize. And if you go back and you read the speeches that they gave, it's quite remarkable. Golgi gets up and doubles down on the reticular theory. Golgi says, I'm right, and Cajal's an idiot, and we'll sort it out later. And Santiago Ramon y Cajal never forgave him. They were bitter enemies to the end. And Cajal was right. Now, odd enough, beyond 1906, because of Golgi's influence in the academy in Italy, and also his influence in politics, he became a senator later on, these ideas, his wrong ideas of reticular theory of the nervous system, were taught well into the 1930s in Italian medical schools, even though they were wrong, simply because of his influence and power. I think it's a remarkable story of the politics of science. It's one of these issues. It was a long time ago, but still, we're not completely immune to politics and science. And this is a great example of it. But let me show you a picture. I really like this picture. This is a picture, for reasons I don't understand, that was on a postcard. And it was a postcard that was sent from Madrid. The image on the left-hand side is a series of drawings of neurons. And the series of drawings from neurons shown here were taken from a pigeon cortex. Maybe one of the pigeons he was feeding in the morning, I don't know, in the afternoon. But you can see these classic examples. Here's a dendrite up here. Here's a neuronal cell body. We'll talk about it in a moment. And this is an axon. Here's another neuronal cell body with this dendrites. This is a pyramidal cell with a great long axon. Now, one of the important insights, in addition to describing the structures of these neurons, and I'll write this out in a moment, but one of his major insights was something known as the law of dynamic polarization. When he looked at these cells, he said information must be flowing in one direction. There must be a unidirectional flow of information, and he said the information is flowing from the dendrite over the cell body to the axon. That was an important insight because it was an idea that the reticulus simply couldn't approach. The reticulus had no explanation for how information would flow in one direction or another if everything was connected by a syncytium. But one of the major views here is that if you have an endedness to these molecules, if they're asymmetric, then we can have a dendrite, a cell body, an axon, and we can get information flowing in one direction. The picture on the right is a picture of Cajal sitting in his laboratory, looking down at the floor pensively. I imagine he's thinking, Golgi was a fool. But let's now take this idea, I'll write this out, and I'll try to explain how Cajal came to these conclusions. And this was the law of dynamic polarization. Now, it's a fancy term, but it says that there is a unidirectional flow of information in neurons. This insight that there's unidirectional flow of information came entirely from the anatomy. At that point in history, they really had no idea of, well, they had a little bit of idea, but they didn't have an in-depth idea of how bioelectricity would occur. They didn't really understand the idea of ion channels. They didn't understand the idea of ions, moving across membranes. But from the anatomy, and in stark contrast to the beautiful drawings that Cajal had, my neurons look like pineapples. But they named these neurons by their structure, and they said the regions out here look like a tree. The Greek for tree is dendros, so they called this dendrite. And the dendrite was characterized by these highly branched structures. So we had all these spikes coming out from the neuronal cell body. They called that the dendrite. Then the region of the neuron where they could recognize the nucleus, this was referred to as the neuronal cell body. In some cases, you'll see the term cell body referred to as the soma. And Cajal recognized that coming out of the cell body was this individual structure, which he called the axon. And that because the cell was a unitary structure, this was the axon terminal. He then said that from this anatomy, he believed there was a unidirectional flow of information, and that the information would flow from the dendrite over the cell body, down the axon, to the axon terminal. And it wasn't long after that that they then began to recognize that there would be sites here called a synapse. Now, Cajal didn't recognize the synapse, he didn't understand the synapse, but it wasn't more than 10 years after his insights that they began to recognize that neurons communicate with one another. At chemical synapses. So now we have this unidirectional flow of information coming from the dendrite to the cell body to the axon terminal. And this functions, we still use this nomenclature, and we can describe this as the anatomic description of the neuron. And the anatomic description of the neuron says dendrite, cell body, axon, and information flows in that direction. But then in the middle part of the 20th century, with the advent of electronics and amplifiers, there was a whole rash of experimentation which began to recognize that these neurons, while dynamically polarized, the new experiments could recognize how ion channels were distributed along the neuron. And this then gave rise to a new description of the neuron, where the above neuron was based entirely upon observation of the anatomy. The new electrophysiology could then be used to describe a functional organization of the neuron. So what I'm going to do in this panel is draw pretty much the same anatomical neuron, but now impose upon this the kind of functional description. So we can describe here the functional description of the neuron. Now, in the first pass here, these two are going to look remarkably alike. So out here, as part of our pineapple, see the nucleus, this great long process. So the way we've drawn this is to be exactly the same. But now, with the advent of electrophysiology, these parts were given new names. This region out here, which includes the dendrite and the cell body, was called the input region. This region here, which extends for these long distances, this was called the conductile region. And this region down here was called the output region. So dendrite, cell body, axon now becomes input, conductal, output. But this functional description relies mostly on the differential distribution of ion channels. Now, out here on the input region, all over the input region, both in the cell body and on the dendrites, will be axon terminals, or output regions. Of other cells. These axon terminals or output regions are releasing neurotransmitters. The hallmark of the input region is that the input region contains a kind of transmembrane protein channel called a ligand-gated channel. This is a fancy term which simply means a channel opened by neurotransmitters. You can even make this more general and say they're opened by chemicals, chemical molecules. You see very, very little of those ligand-gated ionotropic channels here in the conductor region. Instead, what you see in the conductor region in the conductor region, we find channels opened by charge. These channels have a name. They are said to be voltage-gated channels. Another really important difference between the voltage-gated channels we find in the conductor region is that they open to generate an action potential. And this action potential is said to be all or none. Whenever we describe an action potential, we say it's all or none. Either we get it to fire or we don't, so it's all or none. That's in contrast to these potentials up here that result from the release of neurotransmitter. These potentials here are known as synaptic potentials. Where the voltage-gated channels and the signals they generate are all or none, the synaptic potentials are graded with the number, or graded with how many channels open. Simply put, more neurotransmitter, more channel openings, the larger the potential. That's not true here. Voltage-gated channels are all or none. Either you get it or you don't. Either you fire an action potential or you don't. So the input region can be thought of as a kind of integrative site. The axon can be thought of as a signal carrying structure. And then down here in the output region, we'll find that there are special channels which are voltage-gated. Calcium channels. And that these voltage-gated calcium channels trigger neurotransmitter release. So we come full circle. Out here we put these blue structures on. We said these were output regions of another cell, another neuron. They will release neurotransmitter. The neurotransmitter binds to ligand-gated channels, and the hallmark of the input region is the clustering together of ligand-gated channels, those channels that are opened by a neurotransmitter and produce a potential that is graded with the number of channels opening. These signals then will signal the opening of the voltage-gated channels. That will initiate an action potential that propagates along this axon to the axon terminal, at which point that change in voltage, that change in charge, opens the calcium channel, letting calcium in to trigger neurotransmitter release. And we'll go over all of these things in great detail. But what I want to emphasize now is simply to say that the structure that Cajal described as dendrite cell body axon can be redescribed in terms of the distribution of ion channels and the function of these ion channels. And the idea that the input region is ligand-gated channels whose opening is triggered by neurotransmitter. The axons are voltage-gated channels triggered by other channel openings. And finally, down here, we release neurotransmitter. So hold on to those distinctions between the anatomic and physiological description or functional description of the neuron. The other point I want to make here before we move to the next, if you please put your phone away. It's only been 3 times. So the other distinction I want to make here is that this signaling by the channels occurs all along the plasma membrane. That's all at this interface, this very narrow interface between the cytoplasm and the extracellular world. And that this plasma membrane is only about 6 to 7 nanometers in diameter. This structure, the axon, which is filled with protein, can be upwards of 1 to 2 micrometers in diameter. So this is a very thin shell on the outside of the cell, and that's where all the electrical signaling is occurring. But this cytoplasm, as we've seen earlier, is where we move proteins. So any questions up to here?
So is it like the binding of the ligand to the channel sounds like a shock to the axon?
So the question here is it as though the binding of the neurotransmitter to the channel sends a kind of shock to the axon. In a way, that's right. Because what will happen is we're going to find, and we're going to discuss this a lot, we're going to find the opening of these ligand-gated channels allows for sodium to come in. That sodium depolarizes the membrane. That depolarization is carried to a site here known as the trigger zone. And the sodium here then will start to trigger the opening of the voltage-gated channels, and then once those channels are triggered to be open, then that potential propagates along the axon. Anything else? Up top.
So there's like, I mean, like you get a question from whoever that was. That's kind of what I was at, or thinking, or asking, whatever. Are the neurons surrounded in the solution, say, calcium? So when when the gates open, there has to be a potential change in order for that electric current to.
Absolutely, and we'll come to that. But your point is well taken. That for calcium to come in, there needs to be a gradient.
Yeah.
And all around this cell in the extracellular environment, there's a concentration of calcium that's in the neighborhood of about 2 to 3 millimolar. The inside the cell concentration of calcium is down around 100 nanomolar, 0.1 micromolar. So you have like a 5,000-fold gradient of calcium outside to inside. And the other point here that you're alluding to, and we'll spend a lot of time on this, is that the extracellular concentration of sodium out here on the outside of the cell is in the neighborhood of 130 millimolar. Inside, it's about 10 millimolar. The concentration of potassium on the outside is in the neighborhood of about 4 to 5 millimolar. And the concentration of potassium on the inside is about 110. So again, we have these gradients, and we're going to set up the gradient such that there's always more potassium in the inside than the outside, and always more sodium in the outside than the inside. And those gradients then are going to be important, particularly for the signaling for the action potential. Okay? And we're going to drive this home in great detail.
Okay, good.
All right.
The next question would be, are there pumps to get all that?
Then there are pumps. There are pumps. And oh boy, I don't want to go down this road just yet. Let's wait. Let's wait. All right. Yes. Those are the pumps. So you two ought to sit together. So you're right. ATPases are going to be the pumps. Okay. So now what I want to do is I want to move from the description of the membranes and now begin thinking about what's going on in the cytoplasm. And let's use this one again. We'll use the anatomic description. And this was an idea, too, that was recognized fairly early on. And that is that when they started labeling these cells, they began to recognize that ribosomes and rough ER was confined to the cell body. And we've talked about this earlier, and we'll emphasize it again here, is that all neuronal protein synthesis occurs in the dendrite in the cell body. that's a long-winded way of saying is that we'll find ribosomes here. These are ribosomes. And of course, as we saw earlier, many of these are free ribosomes. But some of those ribosomes are attached to the endoplasmic reticulum. And so in our sort of diagrammatic way, we said that there was this endoplasmic reticulum, and that some of the ribosomes were attached to the endoplasmic reticulum. We labeled this as rough ER, and emphasized that rough ER was the site of synthesis of transmembrane, secreted, and lysosomal proteins. And we talked in some detail about the Golgi apparatus, which is the structure named after Emil Golgi. And we talked about the idea that individual vesicles would be budded off from the Golgi, and that these vesicles would contain transmembrane proteins and proteins destined for secretion. And you'll recall that we could then use the combination of the substrate of the microtubules and their orientation with the plus end toward the axon terminal and the minus end toward the cell body. We could use that orientation in conjunction with the kinesin molecules and their attachment to the vesicles to move to transport vesicular proteins. Transport vesicular proteins at rates of about 200 to 400 millimeters per day. But that left out a description of how we can move these cytoplasmic proteins. And early on, they divided the transport into two groups. They said that this 200 to 400 millimeter per day transport is said to be fast, exoplasmic transport. And they said that these proteins, the cytoplasmic proteins, moved at rates of 0.5 to about 0.2 millimeters per day. So now there's a discrepancy of over 100-fold in the rates at which you move these vesicular proteins and the rates at which the cytoplasmic proteins are moved, divided into the fast exoplasmic transport and the slow exoplasmic transport. I just want to take a couple of minutes here to describe how they sorted this out, because I think they're really interesting experiments. What they did, this was, probably 40 years ago now. They asked the question, can we separate the rates of protein movement in neurons? And to ask and answer this question, they exploited a particular preparation. And I wanted to spend a little time on this because I think it was kind of ingenious. What they exploited was the rabbit retina. And the advantage of the rabbit retina is, first, it was pretty big. And secondly, they understood the anatomy of the retina in pretty good detail, so that they knew, if I can get this erased, I'll draw the retina. They knew that the particular neurons in the retina, and they knew that there was one neuron type that sent a process out from the retina and into the central nervous system. So the structure I'm going to draw here in red is meant to represent a retina. So this is intended to be the retina. And in the retina, there are a subset of cells. There are a whole flock of cells in there. There's one subset of cells I want to tell you about. These blue cells are said to be retinal ganglion cells. What's important about the retinal ganglion cells is that their cell bodies are confined to the retina. But their processes leave the retina and enter this structure here known as the optic nerve. And the scientists who were doing this experiment knew that these retinal ganglion cells, with their cell bodies in the retina, extended processes in the optic nerve, but that they had their output regions in a part of the brain known as the thalamus. For the anatomists among you, this was called the lateral geniculate. We don't really care about that here. But what they knew is they knew where the cell bodies were, and they knew where the axon terminals were, and they knew they had a sizable stretch of axon in between. They could then, because the eye was accessible to the outside world, they could then take a needle and into the vitreous, just in front of the retina, they could then inject radioactive amino acids. Now we know, because the cell bodies are here, we know that that's where the protein synthesis is going to take place. So we know that these amino acids are going to be converted into protein. And from above, we know those proteins now are going to be synthesized in the neuronal cell body, but be transported down the axon. So they could wait a little bit of time for the amino acids to be incorporated into radioactive proteins. And then they could kill the animal. And then what they would do is they would take sections along the length of the optic nerve. So they would cut sections along the optic nerve. And then they could plot radioactivity versus time. And when they did that, they found that the radioactivity, which was generated by protein synthesis here in the neural cell bodies, moved at two distinct rates. They found one rate that was moving at about 0.5 to 2 millimeters per day. And they found another peak of protein that was moving considerably faster. It was moving at the rate of about 200 to 400 millimeters per day. So that told them that they had proteins moving at separate rates. And then when they started to sort down and find out what the proteins were, they found that these proteins were all vesicular proteins. And they found that these proteins were all cytoplasmic proteins. This was sorted out some 40 years ago. And the fast axoplasmic transport on microtubules and vesicles with kinesin and dynein, that was sorted out about 35 years ago. But it's only been in the last 5 years or so that we've sorted out why these two move at distinct rates. And we'll pick that up tomorrow.